Alrighty, so for the last section of chapter 4, this is going to focus on organelles that have to do with energy and structure of the cell. So we talk about the mitochondria, and we will spend an entire chapter on the mitochondria. It carries out a process called cellular respiration. Again, these organelles, remember, these organelles are all in eukaryotic cells. The only organelle that we would find in a prokaryotic cell is that ribosome because it doesn't have a membrane around it. Cellular respiration converts chemical energy, so it takes the energy in food and converts it into energy that we can use in the cell. That energy that we use for most of the processes in a cell is ATP. The mitochondria have two internal compartments. They have what's called the intermembrane space, which is a narrow region between the outer and inner membrane. And it has what's called the mitochondrial matrix, which is the very inside of the mitochondria. Now, mitochondria also contain their own DNA. So remember how I said that most of the DNA is in the nucleus? Well, we do have DNA also in the mitochondria. The mitochondria also have their own set of ribosomes. Many of these enzymes are made. So remember, ribosomes are responsible for making proteins. Many proteins are enzymes. So they make their own proteins for this process of cellular respiration. So here's a picture of the mitochondria. So we can see you have many mitochondria inside your cell. They do look like bacterial cells because, in fact, at one point in time, we're going to talk about the history here. They were independent prokaryotes. And so we have the outer membrane, the inner membrane, these folds, and we'll get into more of this specific anatomy and stuff in that particular chapter. But so the intermembrane space, that first compartment would be between those two membranes. The matrix would be the middle. Plants also have an organelle that's kind of similar in structure, different function, but similar in structure to the mitochondria, called the chloroplast. Now, the chloroplast job is to take solar energy and convert to chemical energy. So this is where photosynthesis takes place. So we're using light energy to make chemical energy, and that chemical energy will be used in the bonds between carbons into a molecule called sugar, glucose. The chloroplast has also got little compartments in it. It has an outer and inner membrane like the mitochondria. The inner membrane has inside that, we're going to say there is this really thick stroma or fluid inside there. And we also have the chloroplasts also have their own DNA and ribosomes, which make enzymes. We also have little tiny sacs inside. And again, we'll get into more of this detail in this particular chapter, all on photosynthesis. But we have these membranes called thylakoid membranes that form these structures called grana. And so again, I think pictures are so much easier to see this. So moving to the picture. So here are our chloroplasts in our plant cell. We have the outer membrane, the inner membrane. We have that space in between it, the inner membrane space. These here are the thylakoid membranes. Those thylakoid membranes make these stacks called the grana. And the fluid inside here is called the stroma. So you can see this electron micrograph here of a chloroplast, and you can see the stacks of grana. So here's that evolution piece to it. So when we talk about the chloroplast and the mitochondria, we have what's called the endosymbiotic theory. So we know that they both have DNA. They have their own DNA. They have their own ribosomes. And they can basically self-sustain themselves. So the structure of this DNA and ribosomes, when we look at it, are very similar to what we see in prokaryotic cells. And so based on this, we have the endosymbiotic theory that the mitochondria and chloroplasts were billions of years ago, they were their own smaller prokaryotes that somehow got engulfed by these larger pre-eukaryotic cells. And instead of that larger cell eating it, they formed a relationship where each one benefited. And so here we can see where the mitochondria, well, we're calling the mitochondria now, which is a prokaryotic cell, was engulfed. And basically this cell said, oh, because oxygen was on the rise because of these little guys here. These little guys were photosynthesizing and kicking out oxygen. Oxygen is toxic to many cellular processes. And so many organisms, many of our prokaryotic cells that couldn't tolerate the increasing levels of oxygen died. This little guy said, oh, not only can I tolerate oxygen, but it was using it to burn sugar to make ATP. And so by being engulfed into this cell, maybe it said, well, I can help you out with the oxygen. And the bigger cell provided food and shelter for the little guy here. And so they worked out really well. And again, it's not like something that was, I don't want to say like, oh, we decided that we would work together. No, it worked. And because it worked, it survived. And this is the whole concept of evolution. If it works, it lives, it then propagates itself. If it doesn't, it dies, it goes extinct, right? So this process happened a second time. So we still had this whole lineage here would go off into our animal cells. Well, it happened again. That little photosynthesizer got into this cell here. And now not only did the cell, it doesn't even need to eat food now, right? This little guy will get the food from the sun and inorganic substances, and it'll say, I'll make the sugar for you. And then the mitochondria takes the sugar, makes the ATP. And this itself is a very self-sustaining system. And this would give rise to our plant lineage. So those are our two energy organelles. So when we talk about the structure, a lot of people think of a cell as being like this empty, like a water balloon with things inside it floating around. Well, there is a ton of structure inside a cell. So we have these protein fibers called the cytoskeleton that give it its support. It gives it a highway system. Those vacuoles aren't just haphazardly moving along. They're actually kind of on a highway of these, we'll talk about these microtubules. And so we also have some of these structures are going to be used to move cells with either the cilia or the flagella. So we have three types of filaments based on what they're made up of and how some are made up of the same type of protein, but they're just organized differently. So we have microfilaments, which are actin, and again are involved with motility. We have our intermediate filaments, which reinforce the shape of the cell and anchor it. And then we have these big microtubules that tend to act as kind of a highway system for organelles to move on. Here's a picture of those three types of cytoskeletal components. So we have our micro, our intermediate, and then our tubules here. And you can see how they are organized different. They have used a fluorescent dye to identify those three different structures. And you can see the cell is full of them. So like I said, the cilia and flagella are going to use to help move certain cells. The cilia, we have cilia in our body. We have it in our lungs that helps to push the debris. Here, let me show you a picture of that. There's some cilia there. We use it to push debris up out of our lungs. Females have cilia in their fallopian tubes to move the egg along the way. And then we also have the flagella, which are long whip-like structures we see on bacterial cells, or in animals, we see them in the sperm. So when we talk about the flagella, like I said, it's long, it will whip back and forth, versus the cilia, which are more shorter and kind of whip very quickly to move things along the way. So they act like oars on a boat. Although they are different, and the one is short and stubby and move like oars, and the other one's long and whips back and forth, they do have some common structure to them. And so when we look at it, they both have these microtubules wrapped in an extension. They are extensions of the plasma membrane. We have what's called a ring of 9 microtubule doublets surrounded by a central pair of microtubules. So we tend to call this a 9+2 pattern, and it is anchored in what's called a basal body. So this is in both the cilia and the flagella. So if we have a picture here, you can see these 9 doublets. And again, these doublets, these are proteins. So 9 pairs of doublets with this circular, that are kind of like the spokes on a wheel around these 2 central pair. And then we have these little feet called Dynon feet that act as motor, not really a motor protein, because that would be down here in the basal body here, the base of the motor protein of the cilia or flagella. But they will contract and relax to bend the flagella or the cilia back and forth. So we have, like I said, these motor proteins are called Dynon feet that attach and will slide back and forth. There are, if you Google it or go on YouTube, there are tons of computer animations showing this process moving the flagella or the cilia. So here's a little picture showing those feet, how they bend, and they're going to move back and forth. We have this little segment out of the flagella tail. The extracellular matrix of animal cells also are going to have, the extracellular matrix is like that right outside of the cell membrane. They are also going to have quite a bit of structure associated with them too. They help hold the tissues together. You want your cells to stick together to form tissues, and your tissues to stick together for organs, and we can keep going bigger and bigger here. But it protects the plasma membrane by keeping it from drying out as well. So it's going to protect from wear and tear, drying out, and hold the tissues together. The extracellular matrix, or ECM, is also, we have what are called glycoproteins. These are proteins that have sugars on them, and they bind to proteins called integrins. Integrins will be proteins that will literally go through the cell membrane. So here's a picture of that extracellular matrix. So here's your phospholipid bilayer. Here we have what we would refer to as integrins, which, as I just noticed here, I'm going to do this. All your labels here are in white, so you can't see them. All right, let's go here. So the glycoproteins would be all of these things out here. They are proteins that have sugars on them. There's some more here. There we go. All right, much better. All righty, so I wanted to make sure I have this. You might want to fill them in on the student one as well. It's in your book as well, but I wanted to make sure it's available for you. So again, we have all these glycoproteins. These are all sugars that are kind of hanging off of the proteins. These, we have these long, stronger collagen fibers that help hold those tissues together. Some connecting glycoproteins. These are the integrins that go through. So they're going all the way through the membrane. And then we have those microfilaments here that are going to anchor those integrins. So we have anchoring support on both outside and inside the cell membrane. All right, so we can see that here. So there is a ton of structural support associated with cells. There are 3 types of cell junctions that we're going to find in animal tissues. And so we're going to talk about tight junctions, anchoring junctions, and gap junctions. So tight junctions are going to prevent fluid from leaking in between cells. Certain places we do not want that happening, like in your digestive system. You don't want to start leaking fluid out of your stomach or your intestines into your abdominal cavity. That would be bad. We also have anchoring junctions that are going to help hold those sheets of cells together, and gap junctions that are also going to then allow for flow or information to pass from cell to cell. I have a picture of all three of these. And again, I'm going to have the same problem because of the format. So I am going to go ahead and fix that as well. There we go. So the first one up here are our tight junctions. I think of them as like kind of like a Ziploc bag. If you've ever had a Ziploc bag that won't zip and you pinch it in a couple places to get it started. So that's what I use as my little analogy for that. And so this is going to prevent, this is a cell. These are cells in your small intestine. These are called little villi that help increase surface area for absorption. So your food, your digested food that you have would be in this space up here. And so we want the food to go inside the cell. We do not want the food to go between the cell. If you have food that goes between the cell, it's going to leak out of your digestive system into your abdominal cavity. You may have heard of this called leaky gut syndrome, especially with people that have problems with gluten. When people have gluten intolerance and they talk about celiac disease, the cilia here starts to get worn down or filled up with gluten, so it doesn't do its job. And then we start leaking between the cells. It's not, it could lead to serious medical problems. So here's our tight junctions. Here's anchoring junctions. Anchoring junctions are going to, I think of them as like snaps or buttons on a shirt. And as you know, it still allows your shirt to move, and that's important because your intestines will contract and move to push food along it. So it needs to be able to have that mobility but yet stay intact. So these anchoring junctions will do that. And then we have gap junctions that are like little tunnels between the cells that are going to allow different chemical messengers or ions, and they're very specific for what they will allow to pass through. Sometimes they will be open, sometimes they will be closed. So this allows for cell-to-cell communication. So those are the three different types of cell junctions that we have. Now, plant cells are a little different because they have what's called the cell wall. And this is going to also give the cell more rigidity and protection because we have bones that help give us our structure, and as you know, plants do not. So they have the cell wall that's going to do that for them. The cell wall is primarily composed of what is called cellulose, and we'll talk about cellulose in our biological macromolecule chapter. Now, in order to be able to communicate, because they are multicellular organisms, and if you have that wall in place, you have to be able to communicate from cell to cell. And so what they're going to have are they're going to have what are called plasmodesmata. So here's your plant cells, and they are tunnels. They are just strictly tunnels. Like, so you can see the little lines here, and we'll see those in one of our labs. They're like little straws that go in between the cells. They are not selective. If the molecule or particle will fit, it will move through each one of these cells, which is why it's kind of dangerous for plants to get a bacterial or a virus, because once it gets into a cell, it then has full access to the plant through these plasmodesmata. So this last slide here shows you it kind of summarizes all of the organelles that we talked about in this chapter. Our genetic control, our manufacturing distribution system. Remember, this was also the endoplasmic, the endomembrane system. Sorry. Our processing, our organelles that I talked about, energy, and then our structural support, movement, and communication organelles. So that is the last slide for this presentation, and it ends chapter 4. Again, if you have any questions, feel free to reach out and send me an email, and I will get back to you as soon as possible. Good luck studying, and remember to take your quiz.